So before I start, I would like to greet the organizers and the audience in Ukrainian language. Forgive my accent, please. Я вдячна за запрошення брати участь в літній школі. Я захоплююсь організаторами, які показують приклад патріотизму, прихильність до науки та прагнення до прогресу. І саме головне, будують майбутнє. Hope it's okay. Okay, now let's start. So the title of my presentation is Phages in Our Life and Science. I tried to summarize what I know about this and to share with you. So first of all, let's discuss how many phages are in the environment on our Earth. The estimated number of phages on our planet is about 10 in power 31, which is much, much more than the stars that we see on our sky. Because the stars that we see, the estimated number of the stars is 10 in power 24. But despite of this high amount of bacteriophages around us in the waters, in air and everywhere, only 14,000, about 14,000 complete phage genomes have been listed in the GenBank that cover 30 bacteria genera. This picture here shows the drop of water. Of course, there is a luminescent dye included into this water, and this you can see the big spots. These are bacteria, and around them you can see many small spots that are bacteriophages. So you can have an impression about how many phages are surrounding us. And what are the first evidences about bacteriophages? The first evidences were practically found in the 19th century, in the end of 19th century. There was Ernest Hankin, who was the English doctor who worked in India, and his visit to India coincided with the outbreak of cholera. And he observed people that were getting into the river banks and for these religious procedures, and he was interested why people were doing this, and so he decided to take a sample of water in the beginning of this of the city and in the end of the city. And for his surprise, he found out that in the end of the city, where he expected to have large number of cholera, Vibrio, there were much smaller amount of these bacteria than in the beginning of the city. So he was interested what was the reason for that, and he filtered this water and dropped it on the top of live cholera bacteria, and bacteria was lysed. Unfortunately, this person could not explain this evidence, and it is still known as Hankin's phenomenon. Later on, Nikolai Gamaleya, who was Ukrainian scientist who worked in the Pasteur Institute, he also observed the self-lyse of Bacillus anthracis, and he was the one who published about this in the Russian Archives of Pathology and Clinical Medical and Bacteriology already in 1898. Then after that, Frederick Twardt and Felix Terrell almost simultaneously discovered bacteriophages, but Terrell was the only one who declared that it is possible to use bacteriophages for treatment of humans, animals, and plants. And of course, this was a great, he was great courage to say so, because at that time bacteriophages were invisible and nobody could really trust in existence of these small live organisms. One more person who also contributed to this investigation was Georgian Eliava, the Georgian professor, who observed the same event of lives of bacteria, but again he didn't publish anything. But today, what we know about phage reproduction. Bacteriophages are, of course, bacterial viruses, so their life cycle is parasitic and they need the host bacteria to reproduce their genome. There are several stages of phage bacterial interaction. So one of the first stages is attachment to the surface of bacteria, after which bacteriophage introduces or injects its DNA or RNA into bacteria, where this DNA is replicated and starts to reactivate bacterial host DNA and starts to synthesize its own proteins, after which the proteins and DNA are reassembled again, and the new progeny of bacteriophages is developed, which is pushing the bacterial cell wall from inside, and it opens up, and after that, this progeny is released and starts to chase the new victims. So this is an electron microscopic image of the attachment of this first phase, and here you can see the progeny that is already assembled inside the bacterial cell and is ready to lyse bacteria and to come out and start the process from the beginning. As I've already mentioned, Felix Terrell was the only one who declared that it is possible to use bacteriophages for therapy and prophylaxis. And already in the beginning of the 20th century, he started his experiments. And first people whom he administered bacteriophages was himself and his family members, whom you see here on this photo. This is his home laboratory where he used to work. And the first clinical trial was performed in 1927 by the doctors, in 1921 by the doctors Richard Prunier and Joseph Mason, who published their work, and you can see it here. And the first mass application was performed by Felix Terrell himself when he was in India, Calcutta. And again, there was an outbreak or epidemics of cholera, and he was the people were denying to get bacteriophages. So Felix Terrell put the bacteriophages into the wells from which these people were drinking, and he could get a positive outcome of this. He seized the epidemic in those villages. George Eliava was sent by the independent Georgian government to Pasteur Institute in Paris in 1919, where he heard about a strange guy who was trying to persuade people that there are some live organisms that are killing larger organisms such as bacteria, and the academics from the Pasteur Institute were even trying to make mockery on phylloxera, but George Eliava was accepted all this information very seriously, as he has seen something, some kind of self-lyse of bacterial cells, and he applied to Professor Emil Roux, who was director of the Pasteur Institute at those times, and asked him for the permission to perform the same experiments to prove or disprove phylloxera's use. And fortunately, he managed to prove those experiments, after which phylloxera was invited to the Pasteur Institute, and Darrell and George Eliava became great friends and colleagues. And due to their friendship and collaboration, our institute was developed, which is 100 years old today. Unfortunately, George Eliava's fate is very tragic, because in 1937, like Rutko from your country, he was also executed, and his work could not continue. And phylloxera couldn't move to Georgia as he meant before. However, the experience was developed because of the historical events that took place after 1937. Already in 1939 or 1940, the Finnish campaign had started, and Russia was not prepared for such an amount of wounded soldiers. Therefore, the healthcare organizers decided to use whatever was available to prevent amputations and gas gangrene, which is very common disease during the war. It is called war disease. And you see here the old bacteriophages that are kept in our museum. This field trial revealed positive results, because those soldiers that sorry for my voice. Those soldiers that followed the instructions and applied bacteriophages on their wound immediately as they were wounded, they survived from amputations. This was very good result, of course. And later on, phage therapy was also used during the World War II. And there is a very interesting story about Zinaida Yermolieva and her experience in the Stalingrad. She was a person who was working with cholera bacteriophages, and Stalin sent her to Stalingrad, where there was an epidemic of not only cholera, but also other intestinal diseases, such as dysentery and so forth. So when she was traveling to Stalingrad, the train was bombed, and so she had to go to Stalingrad without any bacteriophages, but she managed to organize underground laboratory in one of the cellars of the ruined building, and managed to manufacture enough amount of bacteriophages, which were given to civilians, and not only to civilians, but also the military personnel. And it is possible to say that the Stalingrad's battle was the fate of Stalingrad's battle was very much dependent on bacteriophages. For this heroism, Zinaida Yermolieva was awarded by the governmental medals. But at the end of World War II, when America and USSR decided to become allies. There was a delegation from British American delegation that arrived to USSR, and this was already after discovery of penicillin. And one of the participants of this delegation was Howard Florey, who later was awarded a Nobel Prize for discovery of penicillin. And he met Zinaida Yermolieva. They exchanged their experiences. And after that, when he was back, he said the following: the stories about phage therapy look as scientific impressionism. So, of course, this was a sentence for the future development of bacteriophages in the West. And the world, we can say, that was divided into two parts due to Iron Curtain. The Soviets continued to use bacteriophages in the medical practice, while antibiotics were promoted in America and Western countries. In the Soviet Union, of course, there was a big progress with the use of bacteriophages, and one of these highest achievements is development of Staphylococcus intravenous bacteriophages, which was used during about 20 years among children and adults, and it decreases the mortality of children, of babies with sepsis. And the pioneers, so you can see these people, these are the pioneers of bacteriophage research. This is Teimur Chanishvili, my uncle, and these are Guram Vasali, a surgeon. This is Bacteriophage, and this is Vakhtang Bochorishvili. And what was happening? What was happening in the West in those years, which we already discussed. In the 30s, Ernest Ruska invented the electron microscope that gave a possibility to visualize bacteriophages. And finally, the discussions about whether bacteriophages are live organisms or are just enzymes, which continued for a long time, so that there were a lot of debates about this, and Felix was very annoyed by this fact. Finally, bacteriophages were recognized to be live organisms as they were already seen in the electron microscope. But what is interesting about this, Ernest Ruska was awarded Nobel Prize in Physics in 1986, only after 55 years after his invention. Later on, Miloslav Demarek and Ugo Fano, these are two scientists that came from different fields. Miloslav Demarek was a geneticist who was working on Drosophila genetics, and Ugo Fano was a physicist who was working on irradiation, ultraviolet, and other types of irradiation, and together they decided to study mutagenesis, and Drosophila was a very sophisticated organism for this, so they decided to use bacteriophages. And for that reason, of course, they have to isolate bacteriophages that are known today, T bacteriophages. Even bacteriophages are the most known. T4 especially is most known because it's not only morphology is known, but also the whole complete genetic map was developed. And you can see that the forms of these bacteriophages is very different. Some of them are having flexible tails, some of them do not have tails at all, and some of them have rigid tails, but these rigid tails are very important because these are those exactly that can work like like inject DNA into the host. Today we know that there are more morphological types of bacteriophages, and Hans Wolfgang Ackermann was the first person who performed classification of bacteriophages according to their morphology, and according to his classification, certain bacteriophage families exist, which include double-stranded DNA that is enveloped, and also single-stranded RNA and double-stranded RNA types, and single-stranded DNA types of bacteriophages. Today, this morphological classification is changed, and all bacteriophage classification should be based on a sequencing, genome sequencing, and the initiator of this classification is Andrew Kropinski. And today, it is the virus taxonomy is currently the responsibility of the International Committee of the Taxonomy of Viruses, and according to the recent data of 2017, there are about 14 subfamilies, 204 genera, and 873 species. Today, Evelyn Andreasen is the head of this committee, and every year it is updated. The details are updated, and probably there are more subfamilies and more genera today, and more species. Now, one of the best probably discoveries for visualization, not only visualization, but also multiplication of bacteriophages, was the plug agar method, which is known as double layer agar method, which was developed by Andre Gracier, who used the lower agar with a concentration of 2%, and the soft agar with concentration of 0.7. And this allowed him to visualize. You see, you can see the plugs. These are exactly those events that are occurring when bacteriophage is lysing bacteria. This is the bacterial loan, which is, you can see here, this is the loan, and the spots that you see, these are the bacteriophage plugs, and it is considered that each plug means one bacteriophage infecting bacteria. This way we can enumerate the number of bacteriophages, live bacteriophages that are present in that or this sample. And also electron microscope gives us a possibility to visualize what we have here and see whether our preparation is pure or not. And this one is not pure. We see two bacteriophages that look like T4 and bacteriophages like this that look like T1. So the further purification of bacteriophages is necessary. Max Delbruck was very excited when he saw these plugs, and in one day you can, he said that in one day you can enumerate bacteriophages. So after this, Max Planck decided to establish the phage group at California Technical University, and in this group there were many people, many famous Scientists like Jan Wiegel from Poland, Simon Benzer, Eli Wollman, Max Delbruck himself, Günter Stand, and others. And these courses, like your summer school, took place annually in Cold Spring Harbor, and starting from 1945, a lot of generations of genetics inculcate with Delbruck's view of how science should be done, quantitatively and critically, were trained. And among them was also James Watson, who is known for the discovery of DNA structure. James Watson has earned his PhD by working on the bacteriophage-related project in the Salvador Luria's laboratory, and the title of this project was The Biological Properties of X-ray Inactivated Bacteriophages. He got his PhD in 1950, and later the Nobel Prize in Physiology and Medicine, together with Francis Crick and Maurice Wilkins. Sorry, what's happening? Okay, and the next very important discovery was made by Max Delbruck and Salvador Luria. It is called fluctuation test. The test determines randomness of mutations of bacteria. The number of mutant bacterial colonies present at the end of the experiment depended on when the mutations arose. Mutations arising in earlier generations would be present in many descendant cells. You can see here and here. So these are the mutations that arose much earlier than the others, and this is like a jackpot. But the mutations that are developing in later generations can be present as few cells. For example, here you can see these few cells. So this is this fluctuation test is called jackpot exactly because of this. Briefly, what happens if you take culture of bacteria from this flask and you try to put it on the top of the agar, which contains also bacteriophage T1, you will see the even distribution of the colonies. But if you cultivate the culture in different tubes, and you do the same from each tube, you culture your bacteria from each tube, you will see that on different petri dishes, you will have different mutants that are resistant to T1 bacteriophage, and those that have developed resistance to T1 bacteriophage before making this bacteriophage, of course, they are earning a jackpot. Salvador Luria and Delbruck were awarded the Nobel Prize in 1969. The next experiment, which was also performed in genetics by Martha Chase and Alfred Hershey, it's called blender experiment. It is a very very clever experiment developed by a woman who used for her domestic reasons, home reasons, she used blender. So the aim of this experiment was to find out what is the where the hereditary information is coded into the protein or onto the DNA. And for this purpose, Hershey and Chase decided to culture bacteriophages on the agar that contained 35S molecules, radioactive molecules, and of course they received the radioactive protein capsule due to this. After that, and this was, you see there is an attachment. Here's bacteriophage is sitting on the top of the bacteria, and then after this they performed they put the culture into the blender and separated bacteriophage and bacteria. And in the bacteriophages, there was nothing. They couldn't find the radioactivated sulfur. After that, they decided to cultivate bacteriophages on the media that was containing radioactive phosphorus. And phosphorus, you know, is in the structure of DNA, and when they performed the same experiment, they found out that bacteriophages really, indeed, they entered into the bacteria, and bacteria contained this radioactive phosphorus. Hershey, not Chase, Hershey, together with other people with Max Delbruck and Salvador Luria, received their Nobel Prize in 1961. They were awarded for the discoveries concerning the replication mechanism of the genetic structure of viruses. Next interesting thing about bacteriophages discovery and bacteriophage life cycle is done by Eugenie and Elizabeth Woolman. By the way, they were Belarusian refugees that escaped from Russia in the early 20th century, and they worked at the Pasteur Institute as well. The same time when Alex Terrell and George Eliava were there, they were great friends. So Eugenie Woolman was the one who said that in the life cycle of phage, there occurs an alteration between infective and non-infective phases. The non-infective phases are perpetuated in the lysogenic bacteria as part of the hereditary structures of the cell. Infective viruses are produced by or from the non-infective phase. And when they applied the enzyme lysozyme, they artificially break the bacteria, they couldn't see the live bacteriophages inside this. Unfortunately, these people couldn't continue their experiments because Elizabeth and Eugenie were arrested by Germans in Paris and sent to Auschwitz, where they were executed in 1943. But the so-called father of lysogeny is Andrei Lvov. In the early 1950s, Andrei Lvov successfully explained how this process, which is known now as lysogeny, works. This is his chart that he developed himself. Now we have a much better visualization of it. So he said that the bacteriophage genes are incorporated into the bacteria's genetic material, but remain latent until a trigger factor causes new phage to be formed. And this trigger factor may be ultraviolet irradiation or maybe shift of temperature or others. And this lysogeny then gave an impact to understanding of gene regulation and developing of operant theory by Francois Jacob and Jacques Monod. They used lysogenic bacteria to develop the operant model of gene regulation to investigate the cellular regulatory mechanism of the lysogenic life cycle and to infer the process of cellular differentiation in the development of more complex eukaryotes. Now we know that one operand gives the rise to one one protein. And these people, André Lebeau, François Jacob, Jacques Monod, they received their Nobel Prize in 1965 for their discoveries concerning genetic control of enzyme and virus synthesis. I must tell you a little story about my connection with André Lebeau when I was many times 30 years ago. I tried to get the postdoc position outside the former Soviet Union. So I wrote him a letter, and he was very old by that time, and he wrote with his trembling hand back to me that he's very thankful that somebody has remembering him still, because nobody has applied to him for a long time. This was after 4 years, he died, unfortunately. Okay, so transduction. What is transduction? Transduction is the transfer of DNA fragments from one bacterium to another bacterium by a bacteriophage. Bacteriophage is a mediator of this process. Transduction was discovered again by Lederberg, Joshua Lederberg and Norton Singer, because why I said Joshua Lederberg again by Lederberg, because he was also discoverer of plasmids. The experiment was very interesting. There was a YouTube, and on both sides of the YouTube were different auxotrophic bacteria, Salmonella, and there was in between of these two branches, there was a filter placed through which bacteria could not move. But amazingly, this strain LA22 that was lacking the possibility to synthesize phenylalanine and tryptophan, but it was able to synthesize methionine and histidine. Suddenly appeared to be prototrophic, which means that on the plate that was not containing the these elements, phenylalanine, histidine, methionine, and tryptophan, these bacteria managed to grow. This was strange. And these scientists, they concluded that something that was much smaller than bacteria could penetrate from one side to another side through the filter and communicate with another host. This was bacteriophage, of course, communicate with another host and render the new host a possibility to synthesize the missing substances. It is interesting to mention also the contribution of Esther Lederberg. Esther Lederberg was the wife of Joshua Lederberg, who developed the method, it is called replication method, She used a velvet, sterilized velvet, of course, and it was possible to make a replica on this velvet, and after that use other plates with the selective media, for example, containing T1 bacteriophages or maybe something others. But she did pioneering the work in genetics. She was the person who isolated the lambda phage. The lambda phage is one of the most famous phages after T4. And the Nobel Prize, of course, was given to her husband, but not to her. And next, very interesting story is about the bacterial defense system, which is called restriction modification system. And first, Luria, Salvador Luria, has noticed that something like this has been happening. And Werner Arber also postulated that bacteria contain restriction enzymes with the capacity to recognize and bind to recurring structural elements of DNA. Then Hamilton-Smith purified this restriction enzyme and showed that it could cleave for foreign DNA. And he also determined the chemical structure of these regions of DNA. And then the third scientist, also Nathan, Daniel Nathans, pioneered the application of restriction enzymes in genetics to develop the restriction maps. This was a big progress in genetics. So briefly, what happens when bacteriophage is introducing its DNA, bacteria is producing the enzyme that cleaves this DNA and thus survives from bacteriophage infection. But at the same time, bacteriophage can modify its DNA. For example, it can use some methylation process, and in this case, bacteriophage can infect and lyse bacteria back again. That's why it is called restriction modification system. This is like a prey and predator. One changes, another changes, one changes, another changes. And this is like a wolf and rabbit. The Nobel Prize was earned by Werner Arber, Daniel Nathans, and Hamilton Smith in 1978 for discovery of mechanisms by which the bacteria resists infection by bacteriophages. And this research led to the discovery of restriction enzymes, which then proved essential for the development of molecular biology. Nobel Prize in chemistry was given to, in 2018, was given to Francis Arnold, George Smith, Gregory Winter. George Smith was the one who discovered the phage display phenomenon. He used very interesting phage, which has no head, but it has it looks like a rope or something like this, and because of this, it has a lot of proteins on its surface. So it is possible to modify its DNA so that on the surface, expression of different proteins can be achieved. And in this case, this means that these proteins will work as antigens, and we can develop a lot of different monoclonal antibodies due to that. And this method is now used for in many diagnostic as a diagnostic tool in many different areas, including medicine, and not only medicine, but also, for example, it is possible to use it for discovery of oats or heavy metals and so forth. And CRISPR, this is very interesting and very novel method. This is clustering regulatory interspaced short palindromic repeats. This is CRISPR abbreviation derives from this. CRISPR-Cas system is a version of adaptive immunity for bacteria. Bacteria retains genetic memory if this bacteria in the past has encountered whatever, I don't know, bacteriophages, antibiotics, and whatever. And it produces complementary small RNAs that direct Cas9 locus nucleases to cleave the same phage DNA if this DNA is encountered again. Cas9 activity is not limited to phages, as I mentioned already, and it is possible to utilize it, different guide RNA to complementary to different genes of interest. So this CRISPR-Cas technique is now widely used for human therapy, genetic therapy. And two women, Jennifer Doudna and Emmanuel Charpenter, showed how the defense system could be turned into cut and paste tool for editing gene sequences. And there is a patent for which two different universities were fighting for a long time. And in 2018, pivotal CRISPR patent battle was won by Broad Institute, where Charpenter is working. The main categories of genetic edits can be promoted with CRISPR-Cas9 system. This is gene inactivation, deletion, or insertion. Insertion in cases of correction of genetics. And now let's go back to phage therapy. In September 2016, the UN meeting tackled the fundamental threat of antibiotic-resistant superbugs, and all UN member states have agreed that it is necessary to take some steps to combat the drug-resistant infections, because the estimated number of people that will die each year will be 700,000, and this is much more than the people that are dying today with cancer. So, but there is a but. This is because phage therapy was developed in the former Soviet Union. As I said, the Iron Curtain was dividing us, and FDA has developed its regulations for the use of antibiotics and bacteriophages. Since they are they are developing, their evolution goes on very fast. Of course, it is they cannot fit into those regulations, but at the same time, people are dying. What is the how can we help? And so there are already many cases when the parents, relatives decided to use personalized phage therapy to those patients that were suffering with different bacterial diseases, and these diseases were untreatable with the modern antibiotics. And one of the stories is about Tom. If you Google, just Google story of Tom. And this guy was traveling in Egypt, and suddenly he got diarrhea, and that looked like a mild diarrhea. And after a while, he got severely sick, and finally he went into coma. And for 3 months, he was in coma. And during this time, his wife, who was microbiologist, working at a very high position in America, she said that I want to save my husband, and I will apply phage therapy. So this was just common Klebsiella bacteria. Phages were developed against Klebsiella and were given to him intravenously and also locally. And he woke up after 3 months of coma. And the first question that he said, what was happening while I was sleeping? And his wife said that the virus from sewage helped you to wake up. And another thing was that President, what was his name? I forgot. The previous president was elected. Another interesting story is about this little girl. She is a patient with cystic fibrosis, and she had to overcome lung implantation operation, but unfortunately it was unsuccessful because she developed infection with Mycobacterium smegmatis. And the only person who works on this bacteria and develops phages against this is Graham Hatfield, whom you see here. So the phages were developed and this girl was saved. This is another story which was popularized, and this will help, I hope, to change the FDA approach to phage therapy. In Europe, the Belgian scientists are working very hard to introduce bacteriophages into the medical practice. And there is Jean-Paul Pirnet, who works in the Queen Astrid's Military Hospital in Brussels. Two of my former PhDs are also working with him, Maya Merabishvili and Thea Lonti. And so his idea is to use magistral phages. And this, what is the meaning of this? It means that, like, you go to the pharmacy and the doctor prescribes to you a certain mixture which contains some different chemical substances. The same way, it is possible that the doctor prescribes bacteriophage mixture containing, let's say, Staphylococcus and Streptococcus bacteriophages. And it's given, it is practically the same as personalized therapy, but more sophisticated. On the way of exceptions of bacteriophages, COVID contributed a lot. And how and why? Because COVID was such a big problem in the world that it was necessary to accumulate all knowledge that was possible. So there were several directions to use bacteriophage against COVID. One of them was that the phage that is not infectious and is made of 180 identical proteins that are placed identically at the trivalent receptors of the surface of this virus. So they have the common receptors. This created an idea that it was possible to use bacteriophage which would mislead the COVID virus, and it would attach to the surface of bacteriophage instead of lung surface. This idea was developed in the Leibniz Institute of Molecular Pharmacology in Germany. Another idea was to use the bacteriophage DNA-based nasal spray with the immune activation against the virus. This was developed in Canada. And the third way of use of bacteriophages against, not directly against COVID, but to secondary respiratory infections, really was implemented. The FDA gave a company which is called Bacteriophage Therapeutics a green light to expand access to use phage therapy to COVID patients which are in the critical state and which were on the artificial ventilation, and they were infected with Klebsiella, Staphylococcus, and Pseudomonas aeruginosa, and it was successful. But while other people and other officials are thinking in the USA, in Europe, in Georgia, we are still working with bacteriophages and applying them widely to our patients and quite successfully. In the conclusion of my presentation, I would cite John Kendrew, who was a Nobel Prize laureate in biochemistry. He said, phages help to make possible an end result which was the most dramatic and rapid development in the whole of biology since Darwin and Mendel. And I hope very much that the situation with the drug resistance will be solved with the use of bacteriophages, either directly by lysing bacteria with bacteriophages or using the CRISPR-Cas system and modifying bacteria back to antibiotic sensitivity. Thank you very much. Дякую.
Дякую.
And I'm waiting.
Thank you very much for your report. It's very interesting. And can I ask you, please?
Yes.
You said about the interaction between the bacteriophage and COVID. as I understand. And
Yes, yes.
maybe, do you know, it is antagonistic activity between these two viruses, or
No,
it
no.
is stimulation of tissue immunity?
No, these are ideas how to use bacteriophages against COVID. Now, one of the simplest ideas was to use bacteriophages against the secondary infections that were developed after COVID. And this was done in America. Another one is that bacteriophages and COVID virus are having 180 proteins, similar proteins. And this gives a possibility to use bacteriophages that will attach to the lung receptors and occupy these receptors, block them so that if you meet the COVID, in this case, COVID will not have a possibility to sit on the same receptors and develop pathogenic process.
Okay, it is.
And the third approach was to use to develop synthesized bacteriophage. So the bacteriophage that is not causing anything. Okay, it is shown that it is absolutely safe. Into the genome of bacteriophage to put the genes that are responsible for the anti-COVID antibody production. Yes. And this way, yes, and use this way bacteriophages. So let's see if somebody will win the next Nobel Prize, I hope.
Oh, it's very interesting. And we have some more questions from our participant, and I read you. One of them from Preludskis, yes, from the Bear history. Do you think bacteriophages can be used to treat viral infection?
No, I don't think so. But of course, it is possible to use the same, as I mentioned, the same idea like I just mentioned. Yeah, this will be not the natural phage, but so-called synthesized phage or modified phage. The genetic tools are giving us a possibility to do so. But there is a problem with any kind of gene-modified organism, as you know, and in general, people are against using gene-modified organisms, you know, the cereals, for example, or vegetables. Mostly there is reluctance to use this kind of thing. So even if such bacteriophages will be developed, I'm afraid that they should go through a very long way to be accepted by the FDA or similar organizations.
Okay.
Natural phages will not. Natural phages cannot be used, yes, to viruses. But what can be done, and this is shown in many articles, bacteriophages are stimulating immune system, general immune system. So this is indirectly the response against viruses.
I see. And one more question, does the phage therapy had bad consequence, and what is the survival ratio? It is from Yulia Ivchenko.
So Yulia, I would like to tell you that the clinical trials are ongoing in different countries. It is difficult to answer your question. Survival ratio, so we are not killing, this is, you can ask this question if we are working with animals, but or other small organisms, experimental organisms, but if when we are talking about patients, we cannot say this. There were 14 officially registered clinical trials, and one of them was the one that we have performed and finished in 2020. This is published in Lancet Infectious Diseases, and you can read there. This clinical trial was performed in the urinary urology clinic. The patients were men that were undergoing so-called TURP operation. This is the operation that is performed when the prostate gland is enlarged for different reasons. And these people had to hold catheters for at least 1 week and longer. So this was our approach, was to introduce bacteriophages to the catheter after the operation to prevent bacterial diseases, secondary diseases that could develop in this case. And what we can conclude after this trial, there were about 100 people involved into this trial, so we can say that these phages are safe. But at the same time, I must tell you that while we were introducing placebo in bacteriophages to catheter, the control group was getting fluorocolons orally for 7 days. So it is uncomparable. Of course, the effect of antibiotics, if the bacteria was sensitive, of course, was overriding the effect of bacteriophages. But in our institute, we have a small outpatient clinic. Every year we have hundreds of people coming from different countries, so from all over the world, I would say. And they usually have urinary problems, upper respiratory problems, or wound problems. These are 3 directions that are in which bacteriophages are used, and we have very good outcome of this therapy, and many of the cases are described and published. So if Julia is interested, she can find it.
And with one more question from Anastasia. Yes, from Anastasia Buziashvili, how it is better to apply phage therapy through the intramuscular, intravenous injection, or pills or spray? It is very interesting.
Anything of that, Gamarjoba, Anastasia. Yes, you can use bacteriophages orally. You can use topically, you can use intravenously, inhalation, any kind of rectally and so forth. But if you decide to use bacteriophages, for example, we are producing commercial bacteriophages that look like they are yellowish, like the media that we are using for production of for growth of bacteria. So those kinds of commercial preparations should be used only topically, orally, rectally, and this way. Or for example, if you put the drops into your throat, yes, you can use it also as a spray. An inhalation. But if you decide to use bacteriophages intravenously, in this case, we have that intravenous staphylococcal bacteriophage, which is super purified, and it doesn't have any kind of dermis and toxins or whatever is produced after lysis of bacteriophages.
Okay. Thank you very much. And
you're welcome.
One more question.
Chlamydia phages. Yeah. We are not working.
Last years, there's a lot of information about chlamydia phages. Do they have any clinical perspective? What do you think about
Probably
this?
yes. Probably yes, because it is believed that any bacteria has phages or viruses. For example, you know, archaea has viruses.
Yes.
Fungi have viruses also. They are not called phages, but they are viruses. So it is quite possible that chlamydia also has viruses or phages. And they may have, of course, they may have the future. But, for example, we tried to isolate phages against Neisseria, and it was unsuccessful. Very often when you are trying to isolate phages, in many cases, they are not virulent, but they are prophages and lysogenic phages, and it's very difficult to develop them into to use them for commercial preparations, development of commercial preparations. So not everything is successful, unfortunately. But hopefully, yes, hopefully,
Future. In future.
yes, future is yours. And there are already new tools, genetic tools, and probably something will happen better than I can foresee now.
Today we have a severe situation with antibiotic resistance and maybe phage. It is our
and the
future.
Wound, yes, wound development in your country. I understand that you have a very, very severe situation, of course, and see what I have.
And one more question. How do you think, will phages still be useful after 100 years? I don't know. It is from our participant, Mikhail Ivansov, but I don't know why 100 years, maybe 200 and more.
Oh, how old is the guy who asked me this question?
Maybe very, very adult.
20? Are you going to live 120 years? I don't know. But I would love to know. Yeah, I don't know what will happen with phages.
And I think you're very lucky because you associate practice and you associate science. And one more question from me. How do you think, is it more stars in the skies or phages in our planet? How do you think?
What I couldn't get.
How many is more stars in the sky or phages in our planet?
In the visual sky, there are less stars than the phages on our planet. In visual sky, because our universe is without any, it doesn't have the edges. So there are, of course, many more planets and stars, but we see 10 in power 24, while on our planet, the phages, the estimated number of phages is 1031.
So we just now have to study how we can use phages in our lives, and maybe it is because in the background of antibiotic resistance and COVID and all these situations we have, maybe phages can help us.
Well, yes, with the COVID, I don't know how you will use for secondary diseases, of course. For secondary respiratory diseases, you can use the phages. And in case of wounds, of course, you can use the phages very successfully. So I hope I wish you to win. Слава Україні, героям слава.
Дякую, дуже дякую. Thank you very much for your report. It's very amazing to speak with you and meet offline many years ago and meet you online today.
I hope that we will meet next year. We will meet in Odessa. I hope so very much.